Okay, hello everybody. Welcome to our lecture on infancy. Now this is going to be the period shortly after birth and on to about 2 years of age. And we're going to discuss the psychological development and some of the physical changes that are occurring within the infant. Now, when we talk about infancy, historically, developmental psychologists have focused on this period of development, and there's a lot of literature, very much literature out there about infancy. And so my class kind of focuses on this period of development to be kind of historically accurate with where the emphasis has been in developmental psych. Now, there's another push for a focus on prenatal development, and there's another push for a focus on geriatrics and later adulthood. And we'll talk about that a little bit later when we get there. But historically, there's been a big focus on infancy because we want to discuss and answer those debates with scientific evidence. Okay, and so psychologists are going to work to understand infancy so that we can lend evidence to particular versions or interpretations of the nature versus nurture debate. Okay, Harlow and Harlow, 1962, conducted a very famous experiment. And maybe you've seen this experiment already where they had like a terry cloth doll and these monkeys were cuddling up next to it and totally neglecting this wired this wired doll that wasn't made of cloth and wasn't soft and cuddly but had food. And they said, no, we don't want the food. We want the cuddly surrogate mom. Okay, well, that is part of Harlow and Harlow's study, no doubt, but usually that's where people stop. Harlow and Harlow's study is much deeper than that. There are many layers to this study, which really made it quite fascinating and makes it very, very interesting. And there's a lot that can be learned from Harlow and Harlow. So how did Harlow and Harlow's study initially get to that terry cloth mom? Well, what Harlow and Harlow noticed in their lab is that every animal comes into it and has a particular quarantine period. Okay? Animals would come into the lab and they'd be isolated socially for a period of time, and they started recognizing that these animals weren't doing so well. And so some of the questions that they had was they wanted to know about Freudian theory. They wanted to know if Freudian theory was accurate or if it was inaccurate, if there's something else that better explains what's going on with our struggles with relationships, our struggles in our social environment. Okay, so here's some questions that they presented. Do personality disorders develop? Is that possible? If so, what is mother's influence on this? And as we talked about the terry cloth mom, and this study is going to be a lot about mothers, and Harlow and Harlow are going to answer some of those essential questions about mom's role in the development of the offspring. They're also going to, and this is the part that's usually forgotten about Harlow and Harlow, they're also going to discuss and answer questions about peer involvement in the developmental process. So it may not just be mom, it may also be the friends, the peers that the offspring grows up around. Okay, and then the real important question is there a critical period of social development in these macaques? Okay, and these are going to be rhesus macaques. We're at the University of Wisconsin, and their development's 4 times faster than humans. And this is important because Harlow and Harlow want to expand upon their conclusions. They want to extend them to the human condition. Now, as as researchers, you really have to be careful with this. But Harlow and Harlow didn't say that this is absolutely going to be true in the case in humans. They said it's very likely that what we found can extrapolate to the human condition. Okay, so phase one, here it is. They bring in 56 infant macaques. They're totally isolated from the group at birth. They're separated, and this is protocol. This is so that they can maintain a disease-free colony. Okay, that's why there's that period of isolation. In most facilities, it's about 30. It's about a 30-day isolation, unless there's some sort of extenuating circumstance. And then, if the animal's healthy and doing well, you know, eating the food of the colony, eating the food from the colony, and its digestive system seems to accept it, and it's not showing any signs of illness, it's not showing any really bizarre maladaptive behaviors, then it will be group housed. Okay, it'll be placed in with the rest of the colony. Okay, well, they did this, and they were required to keep them isolated for a much longer period of time. I believe the period of time was about 6 months that they had to keep these animals quarantined. And then they ended up placing these animals back into the colony and observing what they noticed. And they noticed a lot of stereotypy. Stereotypy is ritualized behavior that is relatively unchanging. It appears compulsive, and it doesn't really stop. So you'll see non-human primates, they'll sway. Sometimes they'll pluck at hair. They'll just keep plucking and plucking and plucking at it. Sometimes they'll pace back and forth a little bit, and they'll just keep going and going and going. Okay, and they won't stop. All right. And then you can also see self-aggression and self-injurious behaviors, or SIB. To the right, you see an image there. That is an image most likely of a primate that has suffered some self-injurious wound. Okay, so this is self-injurious behavior. This primate may have just continually licked at their elbow there, and then wore a hole in it. Okay, now captive colonies and captive keepers of primates are very concerned about SIB. And so we see a lot of really good things going on now in zoos where the climate and the environment around them is more natural for that species. So they do a very good job of minimizing SIB. In captive colonies of laboratory animals, there's a big push for appropriate enrichment. And what is appropriate enrichment? Well, if you have a social animal, it's not just toys, it's allowing that animal to interact with other members of their species. So this is very important. Now, they also noticed inappropriate sexual behaviors and the absence of dominance relationships. They didn't understand how to be subordinate. They didn't understand how to be dominant. They didn't understand that there was a purpose and a culture of dominance relationships. And for macaques, this is very important. If you don't know your relationship to another macaque, it's going to be a very rough life. You need to understand those relationships. Okay, so the next thing that they want to do is they wanted to see if they could intervene to aid these animals in their recovery. And so they took some of them and they put them in the Madison Zoo, and or the zoo in Madison, Wisconsin. And they put them in there in a group. Okay, what they found was some of these animals ended up passing away. They'd never been in this environment, and they ended up getting hurt, and they didn't make it. Okay, but the ones that did formed an appropriate hierarchy. Okay, so when they were reintegrated in the group, they seemed to do well. okay, after a period of time. They could successfully forage for food so they could eat, but they had very inappropriate sexual behavior, and none of the relationships really ended in appropriate copulation. None of the relationships between conspecifics ever led to offspring. And so they introduced a dominant male into this group to see if that would help things along, and it didn't. Okay, they just didn't know how to engage in that type of behavior. Okay, and then when they were returned, the therapeutic value of this group interaction didn't last. They reverted. They went back to their old patterns of behaviors. Okay, so in comes surrogate mom. So with Freudian theory, there's a big emphasis on mom's importance, okay, and caregiver's importance. If mom doesn't provide what this offspring needs, and needs beyond just food, but what this offspring emotionally needs, everything that it needs, then this offspring is going to be in trouble. This offspring's not going to do well. All right. But there's another theory out there that says that mom may only be there, and this is a behaviorist perspective. Freudian theory saying mom needs to be there. There's going to be these internal conflicts that will never be resolved. And so we have that Freudian theory. Then we have behavioral theory that says we've got mom here for food, and that's it. Okay? Mom's not here for any other reason. All right. Both are kind of on the extremes here. And Harlow and Harlow are going to challenge both extremes a little bit. So they give these infants, 60 infant macaques, access to this wire mother that can milk and feed, or this terry cloth mother. And so what ended up happening is the infants formed a very strong attachment to the cloth mother. However, when placed in a room full of their peers, they had very inappropriate play behaviors, very inappropriate sexual behaviors, and only some defensive responses. So what does that mean? They're not making appropriate dominance gestures. Okay? So we now know that mom's not just about feeding, that mom's about comfort, closeness, warmth, feel good sensations. Okay, so there's something else about mom that is important. Excuse me. Okay, phase 4, total isolation. This is the part, this is the part in Harlow and Harlow's study that becomes very unethical. Okay, the isolation period is shorter now, partially because of Harlow and Harlow's, partially because of this study. So you have 30 days instead of that 6-month isolation. Okay, but here's where it really gets tough for some people. Here's what's not usually discussed about Harlow and Harlow, is that, and I like to think of this as, okay, if we're going to, and I'll come back to this in just a second, but if we're going to go to Mars, right? There's a group of people right now that are training very hard. They won this lottery. They put their names in a hat, sent in their applications and wanted to be selected for this trip to Mars. It's a one-way trip to Mars. They know that they're not coming back. Okay. So if you're going to do that, if you're going to do a one-way trip to any celestial body, to the moon, to Mars, wherever you're going to go, are you going to send a large group of people? Are you going to be so sure of your techniques, so sure of your engineering skills, that you're going to send everybody to Mars? Like 100, 2,000 people, something ridiculous. You're going to send that many to Mars. They may not come back. That these individuals have already been told might not survive. Okay. But if you deem that a trip to Mars or the moon or wherever is important, you still need to send somebody. But you're only going to send a few people. And for the first trip to the first landing on the moon, I think we had 3 people. Okay, and everybody usually only remembers the one. All right, but there were 3. One stayed on the orbiting module, and then 2 went down. Buzz Aldrin and Armstrong were down on the moon. Okay, so but we only sent a few because we knew it was dangerous and it might not work out. All right, but it was important. We needed the information. And the same will happen with Mars, I'm sure. If we go, we'll go with just the minimum number that we need to make the whole thing work in case something really bad happens. All right, well, that's what Harlow and Harlow are doing with these monkeys, to find that critical period of social development. All right, they know that these macaques that they're sending, one male and one female in each one of these dyads, that they're going to send into this period of social isolation may not come back. They may be lost forever. Remember, they're saying this is a critical period. So if they don't get social interaction before a certain period of time, they will never be normal. They're done. Okay, so that's what they're doing here. It's really sad. It's one of those things that it was such an important question that it needed to be answered. We didn't want to send, 10, 20, or 100 macaques down this route. All right, but they did, and they isolated them for 2 years, 6 months, or 80 days. Okay, they left them in a dark room. They were by themselves from birth. Okay, for 2 years, for 6 months, or 80 days. Then they were reintroduced to each other. Well, they were introduced to each other. And they looked at play behavior, dominance displays, and courting and sexual behaviors. Let's take a look at what they found. With the 2-year isolation, there was fear of the conspecific, fear of the opposite-sexed individual. They avoided each other. Okay, they didn't play. They looked like aliens to one another. Okay, we don't even recognize. I don't even recognize what you are. All right, you're foreign to me. Okay, and that was something that was very bizarre because they're of the same species. They're opposite sexed individuals, and they're showing no interest in each other at all for anything, not even as a buddy. Okay, there's absence in defense responses when they were placed with dominant animals. They didn't know how to make submissive gestures, and they didn't know how to court a conspecific. Okay, then at the 6-month isolation, the results were very similar to the 2-year period. However, at 80 days, when they were reintroduced to the group, there was normal play behavior, relatively normal defense responses, and relatively normal courting and sexual behaviors that occurred. So what they're saying here is that critical period lies somewhere between 80 days and 6 months, and you should try to be closer. Early socialization, right? You hear this with if you're an owner of dogs, early socialization. You hear this now with infants. Early socialization is important, and this is why it's important, because there might be a critical period of development. Now, for humans, if we're talking 6 months in macaques, right, as being that period that leads those really detrimental, deleterious outcomes, then in humans, we're talking 2 years, a 2-year isolation period. So before 2 years, preferably closer to a year. I mean, if we're talking 80 days, we're only talking a period of about 3 months. And so 3 months is about a year in human. 3 months for macaque is about a year for human. And so we want that socialization to occur, before a year. All right. Now, the next thing that they did is they wanted to tease apart mother's influence and peer influences. So they took some macaques raised by their moms, okay, and they took some macaques raised in isolation. Then they gathered some data and they took a look at the same three constructs: defensive behaviors, play, and sexual behaviors. Infants that were raised with just mom had normal defensive responses, but they were a little delayed in their play and sexual behaviors. Okay? Mom wasn't teaching those things necessarily. Mom was teaching where you are in relationship to other individuals, where you are in the hierarchy. You also see this in other primate species where mom will shield their offspring from playing with higher ranking offspring or lower ranking offspring. Like, you're not they're in a different they're at a different level than you, and learn how to make the appropriate responses to them. And mom teaches those things. When infants were raised with mom and peers, they saw normal behavior all the way across: defensive responses, play, and sexual responses. Now, here's the very interesting part. When the infants were only raised with infants, other peers, okay, they grew up together. They had normal defensive behaviors, play behaviors, and sexual responses. So that's kind of interesting. Okay, so if we go and we take a closer look at this infant-infant bonding, okay, they're grouped, they're placed in this playroom together, and so they're interacting. They're doing, they have that social interaction, and they tend to work these things out on their own. They don't necessarily need that caregiver there to show them how to produce these defensive responses. They'll learn it from playing around. They'll learn sexual behaviors from playing around. Okay, they're going to get there on their own. Okay, now they still need food. They still may need some other things. They still may get there faster if mom's present. Okay, so what are some of the general findings here? Well, we learned that there does appear to be a critical period of social development. Early socialization is very important for non-human primates and humans. It's very important. Okay, and that period needs to occur for humans within the first year. Within the first year, according to Harlow and Harlow, human infants should have some social interactions with peers. Okay, they should start to interact with others. Okay, some ethical concerns. Well, do we really want to isolate individuals again to validate and verify these conclusions? No, I don't think we do. We don't want to do those things. We want to make sure that what we're doing, the questions that we're asking, are ethical questions. Now, we can look back at Harlow and Harlow and go, how could you send and isolate monkeys for 2 years? What was wrong with you guys? Like, why did you do that? Okay, but you've got to remember, a lot of their protocols at the time were about protecting the animals from biological harm, okay? Disease, that was the biggest concern. And we forgot about, for that period of time, we became very mechanical, and we forgot about the psychological component of life. And that's what Harlow and Harlow are chiming in on, the psychological component of what it means to be a living entity and being on this earth, especially if you're from a very socialized species. So here's another way to look at environmental enrichment. One way is socialize the animals. If they're a social species, they need it. Even more asocial species seem to need a little bit of interaction with other species in some way. So it's kind of interesting. Cats are an example. You used to think domesticated cats were just these socially isolated um, individuals, but they have a social life. It's not as necessarily, well, some may argue differently, but I'd argue that it's not as complex and can't be as complex as as the domestic dog's life, for various reasons. I'd lend neurological evidence to that, and the frontal lobe development and volume, and that would be my evidence for it. Some may argue differently, and certainly we're surprised at how complex their social environments are. You know, domestic cats have a social life, so they need interactions with others, occasionally at least. Here we see a slide about environmental enrichment, and you can notice that living in an enriched environment tends to correspond with healthier neurons, and neurons that tend to form more connections, and neurons that tend to last a little bit longer. All right, so what is a neuron? Well, a neuron is the cell that makes up your nervous system. And what we have, just a few structures I want you to know for the exam. We have dendrites. We have, oh, excuse me. We have dendrites that contain receptor sites for chemicals that will bind to those receptor sites and cause action potentials. We have a cell body, that's the soma, and you're going to see it called the soma, and it contains the nucleus and all the organelles that sustain the vital functions of that cell. Then we have the axon, and you can't really see the axon here because it's covered in a myelin sheath, which is just another type of cell called a glial cell. Specifically, this type of glial cell is called a Schwann cell. And the axon actually extends through those Schwann cells. And that's so that when this electrical signal gets sent down the axon, it can jump from node to node rather than having to travel the entire length. So it speeds up that transmission. And you can see that this might be beneficial if you have a very long neuron that extends down your leg. Okay, if that takes too long to get to the muscle, that information takes too long to get there, then you may be delayed in your ability to move or run away from a mountain lion or something. Okay, and then at the end we have these axon terminals where there are neurotransmitters, and once that electrical signal gets there, the neurotransmitter will be released into the synapse, which is the gap between neurons. Okay, and so if you're looking at this, this is just one cell. There are billions of these in the brain. Okay, some neurotransmitters I want you to be familiar with, and keeping in mind this is by no means an exhaustive list of neurotransmitters, but they are very popular neurotransmitters, and they're well-studied neurotransmitters. So we have a pretty good idea of their relationships to psychological constructs and to things that interest us like motor action or motoric action. So acetylcholine is involved in motoric action. It's going to help with the contraction of those muscles. It's also involved with memory and learning. Okay, well, that makes sense. You know, the muscles need to know kind of what to do. And sometimes they need to know just automatically what to do, like walking. Walking is a form of memory. You don't really think about it, and because it's relatively enduring and you had to learn how to walk, it took time to learn how to walk, and acetylcholine was involved in all of that process. Okay, dopamine rewards learning, emotion, and attention. Dopamine's been called the dopaminergic pathway has been called the reward pathway because anything that you find exciting, exhilarating, releases dopamine. Now, the more interesting part of this is that the dopamine's often released and in anticipation of the oncoming reward. So that level of dopamine can actually be higher before you ever receive the reward, just when you think it's coming. Okay. Now, all drugs seem to play in some way on the dopaminergic pathway. So, and then some are specifically playing on the dopaminergic pathway. For example, cocaine will specifically interact with that dopaminergic pathway, making it, a very addictive substance. Endorphins relieve pain. Thank goodness that they do, because if you're running, a marathon or something, you don't want to feel how awful that is the whole time. And guys, I've never run a marathon, and you know why? Because it's awful. Okay? I run like a duck. All right? It would not be good. Okay? Doesn't mean I can't run. I'm just saying, a marathon. Wow, those guys that run that are really, really interested in punishing themselves. But I am amazed by those guys that and guys, I mean all genders and sexes that can do that. Okay. Now, once again, there are drugs that interact at that level to influence that pathway. But your body will do it too, and that's what runner's high is all about. When you get to a certain level of pain, your body knows that it needs to stop that pain, and so it releases those endorphins. Gamma aminobutyric acid, or GABA, is an inhibitory neurotransmitter. It stops the firing of neurons. Okay, it inhibits their action, which is a good thing. I mean, you don't want all impulses all the time. You don't want activation all the time. You need it to calm down. And there's a drug that plays on that system. It's called alcohol. Okay, glutamate is involved in learning and memory. Norepinephrine is noradrenaline. It's going to increase your heart rate, produce that fight, flight, or freeze response, or is very heavily associated with it. Serotonin is involved in sleep, arousal, mood, and hunger. Okay, and those are neurotransmitters for this class. There are many more. Okay, this is an image of the hindbrain, and there are just a few structures I want you to know for the hindbrain. The cerebellum, which is back here at the back, it's kind of this coral-looking structure. Okay, it's primarily for well-learned motor movement. So when you're learning certain actions, like learning to catch a ball or throw a ball or move, it's usually a lot of processing is taking place in the frontal lobe. Then, after you've learned the actions well, it kind of makes its way back here to the cerebellum. And so a lot of athletes, musicians will do things just automatically. Like it looks very easy to them. It wasn't always that way, though, and musicians will tell you when they're learning a new piece, it can be hell. It can really psychologically be a struggle because they're learning new patterns, okay? And that can sometimes be difficult. But once they have it, it just kind of flows. And that's where it's being stored in the cerebellum. Well, walking for us is stored in the cerebellum. Now imagine that walking is gone at some point for you, and you have to relearn how to walk. How frustrating would that be? Because now you're having to process it at much higher levels of organization when before it was just automatic. Okay? All right, the pons are involved and related to sleep. Specifically, they more or less paralyze the spinal column so that motoric action is not produced while you sleep. That's a good thing. Okay, it keeps you asleep. Medulla oblongata is related to vital functions. Okay, like heart rate, respiration, keeping yourself alive. If there is damage to the hindbrain, it's very difficult to survive because everything that's going on back there is for vital functioning, for keeping you alive. Okay. The limbic system. The limbic system is kind of interesting. It's very interesting. We need to talk about just a few structures, though. The first one I want to talk about is this amygdala. Okay, this amygdala is responsible for fear processing. All right, you can't really understand much about aggression and much about many of the world's conflicts if you don't understand a little bit about the amygdala. Okay, there it sits right there. It's for fear processing. In individuals that are very fear reactive, this amygdala is huge. Usually it's huge. Okay, that's a consistent relationship. Now, a very interesting thing about this amygdala is that these olfactory bulbs relay information coming in through your nose straight back to the amygdala. This is the only sensory system that does this. All other sensory systems send information to this thalamus, this bluish structure under here. Okay, and it's kind of tucked away in the image. But that's the thalamus. We're going to call the thalamus the sensory switchboard of the brain. Touch, sight, all are routed through the thalamus first. Auditory information through the thalamus before it goes to other parts of the brain. But smell or olfactory or olfaction goes through to the amygdala first. So when you see your dog walk up and it sniffs a fire hydrant or a post and its hair starts to stand up on its back, it is already angry and upset before the rest of its brain understands what's going on. All right? And the hope here is that that information gets relayed on fast enough before this animal or individual fully reacts with their fear impulse. Okay, same thing can happen with us. And the story I like to tell, the joke I like to tell is, you go into this Sears or J.C. Penney, or you go into some smell good store, and you're walking around, and all of a sudden you catch a whiff of an ex's perfume or cologne, and you go, "Oh, where are they? Oh no, I got to get out of here!" Right? Because you just smelled a smell that was associated with a very fearful stimulus, maybe, and in that X, and so you're like, I gotta get out of here. The other side of the coin, though, is if you're walking through and you smell like the smell of your significant other, and it brings back those very pleasant memories. All right, just a couple more structures in the limbic system. This hippocampus is important for memory, retention of memories. Spatial memories are stored there. And then this hypothalamus. This hypothalamus is your internal thermostat. It's going to try to maintain homeostasis, a stable, constant state within the body. And then just below that hypothalamus is your pituitary gland. And we're going to talk about that pituitary gland again in relationship to the HPA axis, the hypothalamic pituitary adrenal cortex. Okay, that's what that's called. It's that limbic system structure with those two endocrine system structures that help us produce that fight, flight, or freeze response. Okay, lobes of the brain. Only a few things I want you to know about the lobes of the brain. They're listed there. Vision gets directed through the thalamus on back to the occipital lobe. It gets its first crack at that information, and then it relays the information on to other areas of the brain. Remember, visual information doesn't just stop at the occipital lobe. The frontal lobe's involved in executive functions. It's going to need that information from the occipital lobe to make better decisions. What visual information am I receiving right now, and how can I process it, and how can I plan for the future, and what can I predict about what might occur next? Okay, so all of these lobes are going to share information, and there are pathways, and that's really the more interesting part in modern neuroscience, are understanding the pathways and the differences in the structuring of these pathways between individuals, and knowing how these lobes are communicating. Not just that the lobe has a particular function, but how that lobe shares that information with other structures in the brain. That's the more interesting part. Okay, so what can occur during, and I know this is going back to prenatal development, but what can occur during prenatal development to affect the infant? Well, we talked about some teratogens, right? And Zika is a teratogen. And I'm going to let you read the article and work your way through it. But Zika does a lot of very potentially damaging things to the infant and its future life. If there was a very serious exposure, then it's likely that the infant may develop microcephaly and corresponding microencephaly, which means an undersized brain. Okay, now the problem with this is that parents don't always know if they've been infected. It doesn't really present itself all that well in adults. Zika just appears to be a rash or fever or muscle pain in adults. It's mild flu symptoms. And so it's easy to think that nothing really happened. In monkeys, they've shown that Zika can live in testicles for, I think it was a period up to 6 months. If anybody wants me to check that number, I'll gladly go and check it. But I think it was about 6 months, which, you know, makes the CDC a little bit concerned about humans and how long Zika may reside in human testicles. All right, so it's not just a problem and a concern for pregnant mom. It's also a concern for future potential father. All right, so we are concerned about Zika, and it is in Texas, so we do need to be concerned, especially going into our summer, rainy, moist months sometimes. Okay, radiation and its impairments. And I know this is once again kind of looking at prenatal development, but we want to talk about what's going to happen in infancy. So if there's exposure to radiation within the first 7 weeks, the infant usually doesn't survive or develops microcephaly, well, an underdeveloped head. So it's not just Zika that will do that. There are other things that will do that too. People kind of get this idea that once it starts showing up in the news, that's the only thing that can lead to this outcome. Well, no, there are usually multiple things that can lead to that outcome, which makes it even more tricky to isolate causal relationships. So with that said, Zika doesn't cause microencephaly or microcephaly. It is highly related to it, though. Okay, not all individuals exposed to Zika present symptoms. Now, 8 to 15 weeks, there are usually issues with neuronal migration. You should have watched that video to know specifically what the issues were for neuronal migration. And then there are prenatal abnormalities as well. Then 16 to 25 weeks, usually some intellectual disability and maybe some functional issues in the left hemisphere. And then after 26 weeks, possible intellectual disability. And the images are going to show you different types of radiation. So we can see certain types of radiation as visible light. Once we get to ultraviolet and infrared, we can't see those anymore. Then we get to the other image where we're looking at certain types of light, like alpha radiation usually isn't a problem. It can't penetrate skin, so it's not really an issue. Beta radiation can. Medical x-rays certainly can. They can even go through wood. Gamma radiation can get through cement. Right, so then we get to the more ionizing radiation, which is what we're concerned about. So infrared, we're not really concerned about. Microwaves, radio waves, not really all that concerned about. The ionizing radiation, though, the ultraviolet, the X-rays, the gamma rays, those are what we're really concerned about. Okay, so two different patterns of development I want you to be familiar with: the cephalocaudal, proximal distal. Cephalocaudal is from the top down. Infant's born with a ginormous head. Then the rest of their body starts to catch up. And the eyes are the size that they're always going to be. So many features on the head are already there, and they're not going to change too much. Proximal distal patterns are where we're developing from the abdomen out. So they'll start to get a bigger abdomen, and then their arms will eventually catch up, along with their legs. And their neck. And their neck's a very important one because they can't really keep that big head of theirs stable. So when you put them in something like a moving automobile and there's an accident, well, adult's head has a very muscular neck that kind of protects the spine and the spinal cord. But for an infant, that doesn't happen, and so you get some really nasty things that can happen during a car accident, like an internal decapitation. And I don't want to bring this lecture down, okay? But that is something that can happen if the infant is not properly restrained in the vehicle, is that they could have an internal decapitation where the spinal cord gets severed because their head kept moving, but their body stayed still and they didn't have the neck muscles to stop it. Okay, height and weight. I'm going to let you look over this. Remember that all of these are ranges, so you don't really want to grow concerned about a newborn's height if they're at 18 inches and the doctor says, well, they're a little short. Well, short relative to what? What's the 95% confidence interval? What are the characteristics of the parents? So you just kind of want to keep these things in perspective, and a lot of parents grow really worried about these, but I'm just going to leave it right here and we'll move on.